We will continue with the discussions of the clinical chemistry. So we will talk about the types of the analytic errors or the errors that is happening during the sample. So we subdivided into three types: pre-analytic errors, analytic errors, and post-analytic errors. So we will start about the pre-analytic errors, the errors that occur during the sample collection, transport, processing, which everything that is prior to the analysis processing. It may be that is referred to the test or the patient, the collection, and the transport. So we will start about the test orders, the wrong test that is maybe ordered, the wrong test that is ordered, or the test order misunderstood. If that is related to the patient may be improperly prepared or the patient misidentified. Like if we want to fasting patients for measuring their glucose level. So if the patient come just he ate his breakfast, and that means the patient is improperly prepared. The patient misidentified. Also, that is maybe referred to the collection. It's in the wrong container, or the specimen it's mislabeled, or inadequate amount that is collected, or there is a problem in the transport, like under improper condition, like we didn't do the accurate incubation while we transport this sample, or there is a delay in processing and the transport time is too long, or inadequate centrifugation, because we said that centrifugation we have to make the collection of the sample, it will stay for 30 minutes, and then we just put it into centrifugation for 15 minutes under the rate of 5000 rpm. What is referred to the analytic errors, the errors that is happening during the analysis, during the testing. So maybe the instrument not calibrated properly, maybe that is referred to the instrument that we use it and it's not calibrated properly, or that is due to the interfering substances that is present inside the sample, like we don't know or we don't recognize that there is interfering substances in the sample, and that is lead to the analytic errors. Sometimes that is referred to the dilution error. Like we said, one of the interfering mechanism of the hemolysis was dilution. Sometimes the solution or the sample is really diluted, so we can measure the analyte so specifically. And also the bubbles or the particles present in the sample. So all these errors are referring to the testing. So maybe that is referred to the instrument that is not calibrated very well, and also interfering substances that may be present in the sample and not recognized, or that is referred to the dilution error, or the bubbles and the particles. One of the most important errors that there is exogenous particles or bubbles that is present in the sample. Post-analytic errors, the errors that is happen after the analysis. So like reporting results, reporting inaccurately, like reporting of these results is very inaccurate, or the results sent to wrong location. Maybe the results itself are wrong, or the results sent to the wrong location, or the report delayed or incomplete. So all these errors is called post-analytic errors. So pre-analytic errors, some that is referred to the test order, some that is referred to the patient, to the collection, to the transport. Analytic, it's what is happening inside the sample, and post-analytic, it's in the reporting of the sample. So here we talking about the absorbance, and here the like what are the absorbance of each one. As you can see here, NADH, and we said the absorbance of the NADH is 340 nanometers. This is the absorbance of the NADH. So here the absorbance of it, so we can say it's a 340. And about the hemoglobin, actually it's very range, it's sometimes from, but it's actually between the 400 to the 500. Icterus, it's 400 to the 540. I mean the no, it's the bilirubin. I mean here the bilirubin, it will be from between 400 to the 540, and we here have lipemia. The lipemia, it's between 300 to the 500, all the lipids. Okay, these are the absorbance of most of thing, and we said that the hemolysis, icterus, lipemia, they are pre-analytic errors and they are interfering factors. And we have discussed before what is HIL indices, and we said the HIL indices that is used to measure the hemolysis, icterus, and lipemia, like fetal hemoglobin, total bilirubin, and also lipid. And we said that we measure it in the units like a gram, hemoglobin a gram per liter, bilirubin it's micromole per liter, and the lipid is nano or millimole per liter. Okay, the bilirubin millimole per liter and the lipids micromole per liters. NADH is nicotinamide dinucleotide hydrogen. Okay, so what is it? It's actually, we always said NADH, but what is NADH? Actually, NADH is nicotinamide dinucleotide hydrogen. Nicotinamide dinucleotide hydrogen. So we have the dietary protein and we have also the body protein. So we have two types of the protein, because we now enter another topic to discuss it, and I have discussed it before. It's about the uric cycle. Okay, we have two cycles. We have the uric acid cycle and the urea cycle, and both of them we will discuss in the lecture, but I discussed all the cycle also in isolated form. So we have here the first one is dietary protein, the protein that we take it is through the diet, and we have also the protein that is already present in our body. So we have two types of the proteins, but the problem is not here. Both of them we digest. It happens digestion and metabolism, and that is due to the production of NH3, which is ammonia, in NH4, which is ammonium, and ammonium is a toxic compound. It's more toxic than the NH3. So again, before we go, what is a urea cycle, what is the productions and all these things, just again, we have dietary protein, we have body protein, then undergo into the digestion and metabolism, and that is lead to the production of NH3, which is ammonia, and H4, which is ammonium, and the ammonium is a toxic compound. Then ammonium and the glutamine and this cycle, okay, and all this cycle that is lead to something is called urea cycle, because we know that all of this stuff happened inside the mitochondria of the liver and the cytosol of the liver, the first reaction, which we can say the urea cycle, and that is leading to the productions of urea, and that is exactly the urea. The urea, as you can see, C double bond with the oxygen, we can see like this, and we have bond with the NH2 and H2O. So it's a wasted product because the carbon is already has 4 bonds, so it's not useful. That's exactly why we said the urea is a wasted product, because if the carbon has that is can bind to another thing, then it's not a wasted product. So the urea here, because the carbon has double bond with the oxygen and 2 bonds with the NH2 and H2, so it's a waste product. It's a waste product because now the carbon is oxidized. Here is oxidized carbon, so it's useless, so it's a good to rid of it. Okay, it's useless now. And because if there is a carbon without, if there is a carbon but there is no oxidation, it's important to produce energy, so it's not a wasted product. So it's actually a long journey of the urea cycle, and that's the most important thing, but I have just discussed it on the isolated YouTube. I will also in the descriptions of this video you will find all the links that is associated with the urea cycle, because I explained it in a very detailed form. So actually we have a dietary protein, we have the body protein, then undergo digestion and metabolism that is lead to the production of an H3, which is ammonia, and H4, which is ammonium, and this is a toxic product. And after all this urea cycle, that is lead to the production of urea. The carbon here has a double bond, or the carbon here is oxidized, that why is the urea is waste product. So here the urea is the waste product. Why? Because the carbon is oxidized, so it's good to get rid of it. If the carbon is not oxidized, then we use this carbon for the production of energy. Remember also that the first reaction of the urea cycle happened inside the mitochondria of the liver. The most important reaction, that is this reaction, is happening inside the mitochondria of the liver. So we have CO2 and we have an H3, which is a result from the protein, as you can see here, ammonia. Then with the carbon dioxide oxidized with the help of this enzyme, and we said this enzyme is called rate limiting enzyme. So it's a rate limiting enzyme of the urea cycle. It's called carbamyl phosphate synthase enzyme. Okay, and the phosphate synthase enzyme that is take 2 ATP, and that is lead to the production of 2 ADP plus phosphate, and that is lead to the production of carbamyl phosphate. So this is the first reaction of the urea cycle that is happening inside the mitochondria of the liver. We have CO2 plus NH3, which is ammonia, that is result from the metabolism of the protein, whatever is the source, under the action of the enzyme carbamyl phosphate synthase 1 enzyme, that is lead to the production of the carbamyl phosphate, and it's a rate limiting enzyme. So it uses 2 ATP to produce 2 ADP plus phosphate group. Then there is something, or this reaction need help or cofactor of N-acetyl glutamate. N-acetyl glutamate composed of two forms of a glutamic acid and acetyl CoA. So it composed of two things, because acetyl comes from the acetyl coenzyme A, and the glutamate, because glutamate, it's a glutamate, so it's a glutamic acid which end with the carboxylic group. So the acetyl coenzyme A, it results from the glycolysis and fatty acid oxidation, and its level indicate how energy that is needed. Okay, the presence of the acetyl glutamate, acetyl I mean, and the glutamate, glutamic acid, from the transfer of glutamine to the liver. So that is also produces glutamine, or released from the liver. Okay, so large amount of the glutamine that is released from the liver. So again, acetylglutamate, which is a cofactor for this reaction, actually we called it an acetylglutamate, composed of acetyl coenzyme A, which is result from the glycolysis from the fatty acid oxidation, and that is indicate how many or how much energy is needed, and the glutamic acid, that is results from glutamine that is synthesized by the liver. So acetyl coenzyme A indicate how enough energy have the cell to start the urea cycle, like how much energy have to start the urea cycle, because as you can see here that we need 2 ATP to produce 2, so we need energy to make this reaction start. And when I said rate limiting enzyme, the rate limiting enzyme is the slowest enzyme in whole urea cycle, so it needs more energy. Okay, and if there is enough an H4, that is reached the liver, because NH4 is more toxic than the NH3. So the N-acetylglutamate is the cofactor that is used for the first reaction, and it indicates if it has enough energy or if it has enough NH4. I really explained, I will again tell you, I really explained in a very detailed form the urea cycle, but here I'm just talking about some points about it that we have to care about them, okay, and we have to memorize them very carefully. I will put everything on the description. So the question says here, how many amino acid that is used in the urea cycle? So the question is, how many amino acid that we use in the urea cycle? Actually, we use arginine, and we use also arginine, we use citrulline, we use aspartate, and we use arginine. So we have like here 5 amino acid: arginine, arginine, citrulline, aspartate, arginine, succinate. Maybe we just understand that the amino acid, okay, arginine is amino acid, aspartate is amino acid, but what is arginine or citrulline or arginosuccinate? It's just compositions of amino acid. That's why we called it as arginine or citrulline. So we have or we used all these members like arginine, aspartate, citrulline, arginine, and also arginine succinate. What is the best marker now that is used to measure the kidney function test? Creatinine. So the creatinine is the waste product of the kidney, is the best indicator of how the function of the kidney, or how good is the function of the kidney. So we can't measure it by the urea. It used in monitoring of renal replacement therapy. We measure the urea in the dialysis fluids, for example. Yeah, we that's not mean when I said the best indicator and the best marker for the kidney function test is the creatinine. That doesn't indicate that we can't use the urea. Actually, we use it, urea in the lab test, but mainly or more useful in the case of the dialysis fluid, we measure the urea. In special cases, we measure the urea. But if we just talk about the functions of the kidney, the best indicator for that is creatinine. Why? Because the creatinine is the waste product of the kidney, or excreted through the kidney, and so it shows us the ability of the kidney to excrete it. So if there is an increased level of the creatinine above 1, that's mean it's really there is a damage in the kidney. But the urea, it's an important in the case of the dialysis or replacement therapy. Then we go to the urea analysis. As you can see here, we have the urea, and we said again we have here the urea, and as we see here that the urea is oxidized. Here the urea is oxidized, so it's a waste product, and we have 2HO in the help of the urease enzyme, so that is lead to the formation of NH4 plus CO3. So if we not get rid of this urea by the enzyme, which is called urease enzyme, that is lead to the production of NH4, which is ammonia, and CO3 or NH3 plus CO2, but that is lead to the production of ammonia or ammonium. And then the ammonium and the urease, you know, that is happened by the bacteria. So NH4, that is binds or interacts with the oxaloglutarate by the help of the enzyme called glutamate dehydrogenase enzyme that use NADH positive. Then it lead to the production of the glutamate plus H2O. So by this mechanism, we get rid of the urea and change it to the glutamate. Okay, that is the changes to the glutamate, and that, or by the way, we get rid of urea, which is toxic. Glutamate, okay, it's a glutamic acid, and we turn it by this mechanism, so we need oxaloglutarate. You know, this reaction that we are talking about this here, that this reaction actually is so important because by this mechanism we just get rid of the ammonium. Okay, so we have the urea plus H2O by the urease, that is lead to the formation of NH4 plus CO2, then NH4 plus 2-oxaloglutarate by the help of the enzyme glutamate dehydrogenase, then that is lead to the formation of glutamate and H2O. So about the determination of the ammonium, so we just make a ratio between the concentration of NH4 divided by the concentration of NH3 at the pH 7.4, which is equals 100, because NH3 is more toxic than NH4, so that is good for the metabolism. Sometimes they said the NH4 more toxic, and sometimes they said NH3 is more toxic. So some resources say that every time they both toxic, and that's what's so important about these conditions. Use ammonium free refrigerated tubes with the heparin or EDTA as anticoagulant. Why? To prevent the contamination, prevent the clotting. We know that the EDTA, we also explained that before, and we said that EDTA, that is act as irreversible anti-clotting factors, and that is binds to the calcium and keep the morphology of the cell. This is like a characters of it, like keep the morphology of the cells, make not any kind of the changes to the cell. And it used to measure the blood smear, that is used to measure also skill cell anemia flow cystometry. All that is related by the ETTA. So here we put it in the tubes that is refrigerated, and also that is contains heparin. Also heparin acts as anticoagulant. Also EDTA also acts as the anticoagulant because we want any contamination, any clotting in our sample. Process sample quickly on the ice and use refrigerated centrifugation for 15 minutes, so that is will reduce the metabolic activity and reduce the bacterial growth, because we don't want that the urea is acting. So that is lead to the elevated or falsely elevated because of the hemolysis, and also increase the level of the gamma GGT or bile disease. Mostly we use photometric assay, and we know that the glutamate dehydrogenase is 340. The absorbance of the glutamate dehydrogenase is 340 nanometers, and as you can see here that the glutamate dehydrogenase enzyme, we see dehydrogenase, so the calcification of this enzyme is 1 because it's under the oxidoreductase enzyme, dehydrogenase enzyme. That is lead to urea plus H2O by the help of glutamate dehydrogenase, and we use the NAD H to the NAD positive, so that's why the absorbance is 340. So that is lead to the productions of the NH4 plus CO2, and then the NH4 plus oxaloglutarate by the help of the glutamate dehydrogenase, then that is lead to the productions of a glutamate. So we have some notes about these things, like plasma contains nitrogens more 1000 more than the ammonia. Okay, So failure, prune and mishandles that is lead to the hemolysis, which is converted to the ammonia. Okay, so if we just see the normally that the plasma contains nitrogen 1000 times more than the ammonia, and then if there is mishandles or a prune or something, and that is lead to the hemolysis, that is lead to the production to the ammonia, and the hemolysis increases the ammonium because the breakdown of the RBCs, that is lead to the degradations of the glutamine, and that is lead to the production of amino acid, and that is lead to the elevated level of the NH4, and that is lead to the urea. Okay, NH4 in a normal liver, it will be turned to the urea, but if NH4 in the pathological liver, that is lead to the NH4 because it doesn't work, and elevated level of the gamma GT. Okay, so the hemolysis, if we are talking about the hemolysis, that will lead to an increase the level of NH4. So hemolysis increase the level of the NH4, and we see that the mechanism, because it's destructions of the RBCs, so we causes like destructions of these amino acid and the proteins that is present in the RBCs, so that is lead to the production of glutamine, which increase the level of NH4. In the normal liver, that is turned to the urea. In the pathological, it will stay in H4. Neurotoxicity of the ammonium and H4, which is called ammonium, and that is lead to the accumulation of a glutamine asterocytes because they are just reversible reactions, and these glutamine affect the asterocytes, and these asterocytes, they will be swallowing, so that is lead to the swallowing of the asterocytes and causes a brain edema and the brain stem compression, asterocyte dysfunction, so that is lead to the asterocyte dysfunction. Hyperuricemia. Hyperuricemia, so the other thing is uric acid. Okay, uric acid, because we have to be careful about the urea and the uric acid, because the urea, it comes from protein. Again, just remember what we used. We said that dietary protein, body protein, whatever the protein, and then it turned to the NH3 and H4, and this turned to the urea. So remember that the urea comes from the metabolism or degradations of a protein, but the uric acid comes from a purine, which is coming from the DNA, which is completely differ. So it's called 2,6,8 trihydroxypurine. 2,6,8 trihydroxypurine. Why we said that trihydroxypurine? If you just remember or memorize that the uric acid is 2,6,8 trihydroxypurine, then you understand now that purine, that is lead to the uric acid. And also again, I'm and I just explained that in a very detailed form. The uric acid cycle explained everything about the purine and how it turned to the uric acid, and I discussed also the disease that is related to the uric acid metabolism, and I will also put it in the description of this video. So we have the uric acid. The uric acid is composed of 2,6,8 trihydroxypurine, so it's result from the purine. It's the major product of the catabolism of the purine. So the endocytes in nucleic acid, that is composed of the purine, and the purine that is composed of the adenosine and the guanosine, determined by the bacterial uricase. The bacterial uricase turn it urate to the allanitone by using of oxygen to the H2O and CO2. So from the urate, it will turn by the bacterial uricase to the allanitone, and that decreased the absorptions to the 293 nanometers, which are easily compromised by the proteins urate again by the enzyme is called uricase, and this enzyme that is present in the bacteria that is lead to the allantoin plus H2O2. Then the H2O2 plus oxygen acceptor peroxidase that is lead to the colored compound ureate and oxygen receptors uricase plus peroxidase that is lead to production of the allantoin colored compound and H2O2. Like we just combine the two reaction with the others, like we have the ureate. When I said ureate, that's mean uric acid, and by the help of the enzymes of uricase and then peroxidase. So the enzyme, the first one is uricase and then peroxidase that is lead to the production of the allantoin colored compound and H2O. In the case of the hyper, not uremia, when I said hyperuremia, that's mean the urea increased by hyperuricemia or uricemia because it takes from the uric acid. So that is lead to the production that is lead to the disease is called gout, and the gout that, or I will explain it later, and also the renal stone formation. One of these mechanisms of increasing the level of the uric acid in the blood that is lead to the formation of the leads to the multiple diseases like a gout and the renal stone formation. The gout is the disease that is due to the accumulation of monosodium urate participates in the joint and the tissue, especially in the first appear in the big toe, because the urate precipitate under the low temperature, and which of our body parts has the lowest temperature is the big toe of our feet, so it will appear these symptoms in the first on the big toe. So gout is due to the accumulation of the monosodium urate, and it will like participating on the precipitate, I mean, in the joints tissues from sequestrated body fluid. So the gout that is maybe lead to the complications like ureate nephrolithiasis, so that is lead to the stones in the kidney, and that is maybe lead to the acute intratubular depositions, or maybe lead to the gout nephropathy. So kidney disease is associated with the hyperuricemia, that's mean a uric acid. So the uric acid by the help of the enzyme uricase, okay, because this we want just to know the difference between them. Actually, we have uric acid, okay, and by the help of this enzyme, which is known as uricase, that is lead to the formation of a allantoin, okay, which is not found in the humans and used to treatment of the gout. Uricase is present actually in the bacteria. So we have the uric acid by the help of the enzyme which is known as a uricase, and that is lead to the formation of the allantoin. Okay, uricase is present in the bacteria, it's not present in the human at all. And here also, or we can say not only in the bacteria, but actually it present in all the mammals, in the animals even, but not in the human. Urea by the urease that is lead to the formation of what, NH4 or NH3 plus CO2. Okay, and the urease that present in the bacteria. The compound, its absorbance is 293 nanometers is tyrosine. This is the compound, its absorbance is 293. Okay, then we discuss the ketone bodies. Ketone body. Also, I explained the ketone bodies in separated video about the ketone bodies, and we said that the ketone bodies is subdivided into acetoacetate, beta hydroxybutyric acid, or we can say butyrate, and the acetone. So these are the types of the ketone bodies. So when just the questions, what are the ketone bodies? Acetoacetate, acetone, beta hydroxybutyrate. Nitric acid is the acid that used to detect the bacteria in the urine. So this acid, it just show us if there is a nitric acid that is present in the urine, so that's mean there is a bacteria in our body. So yeah, that's why when just we use the urine sample, and if it present nitrate in the urine sample, then that's indicated a bacterial infection. So the nitric acid, when it present in the urine sample, so that indicates infections. We have also heterophilic antibodies. They are antibodies that use to detect and react with the antigen with a different species. That's why we said heterophilic antibodies, like, if the urea cycle is not sufficient, how can we get rid of NH4? Yeah, to the glutamine, exactly. And by the help of glutamine dehydrogenase enzyme, that it will changes to the glutamine, so we get rid of the ammonium. Glutamine is less toxic and can be transported to the liver and the kidney. How can we treat the neonate with increased bilirubin? As I said, sometimes the neonate comes with an increased level of the bilirubin, unconjugated bilirubin, okay, because of immature liver. So by phototherapy, we can by the liver transplantation, we can blood transfusion or exchange transfusion or IV immunoglobulin or hydration. So that's what we can help the neonate if it has increased bilirubin. But actually, if that is like we can say physiological, yeah, we can use phototherapy, we can use exchange transfusion, we can use also IV immunoglobulins or hydration. Again, these questions again, why the paracetamol is contraindicated in the Gilbert syndrome, because we said there is a reduced form of the uridine diphosphate glutarate enzyme, and that because of reducing the same enzyme that is important for the metabolism of the paracetamol. So if the reduced level, so that is will lead to the accumulation of the aspirin, and the accumulation of the aspirin that paracetamol, that is lead to something what we called it as, yeah, that is lead to the accumulation of the aspirin and the accumulation of the paracetamol, that lead to the increase the toxicity of the liver and destroy it completely. So again, why the paracetamol is contraindicated in the Gilbert syndrome? Because in the Gilbert syndrome, there is reduced level of the uridine diphosphate glucosyl transferase enzyme, and that is lead to decrease the metabolism and excretion of the paracetamol, and then accumulated inside the liver, and that is lead to the toxic liver. It's not okay if I'm not sure about the results, so we make two rating, multiple shake, and sometimes we dilute the sample. D-Rates ratio, also we discussed that before. D-Rates ratio, it's the divisions of AST divided by the ALT, and we said the half time of the AST is 17 hours while ALT is 47 hours. AST more ALT, that indicate alcoholic substance. Yeah, that's mean more severe liver damage, especially alcohol. But if ALT more than AST, that indicate viral infection. Okay, why it's indicate the viral infection? Because ALT it's present more than the liver. So ALT present in the, I think wrong here, because AST is present in the liver, the only in the liver, in the mitochondria of the liver. That's why we said the AST is more organ specific, but it's not a disease specific. ALT, that is present in the liver, in the heart, in the muscle. I think I just reverse these enzyme. Which polar compound is important for the conjugation with the non-polar compound? Yeah, if we have a non-polar compound, what is the polar compound important for the conjugation? Sulfate, glycine, sugar acid. So what does the questions mean? That's mean the sulfate, glycine, and sugar acid, they are all of them, they are polar compounds. We use them. We use the sulfate for conjugation, we use the glycine for conjugation, we use sugar acid also for conjugation. Choline esterase is produced by the liver mainly, and we said that the choline esterase, we have two types. We have the true and we have the pseudo. And the true, that is present in the RPCs, present also in the liver, but the pseudo, it is present in the plasma because it's produced in the liver and then go to the plasma. So in the damage of the liver, it will be decreased. Okay, in the case of the bilirubin, we use actually sugar acid for conjugation because the bilirubin is non-polar. What we can use a glycine, we can use a sulfate, we can use sugar acid, and the sugar acid is the best for the conjugation, but not always. Actually, we use, no, yes, exactly, sugar acid. I mean, albumin is important for the transport. It has no role in the conjugation. That's my questions come so rapidly. How come bilirubin, which is not water soluble, travel to the spleen by the albumin? Why is it here that the GGT and alkaline phosphatase increase in this case? Because the GGT and the alkaline phosphatase, both of them, they are present, are called them membrane bound enzymes, so they are just present in the hepatobiliary system. Once there is a damage to this hepatobiliary system, so the gamma glutamine transferase enzyme increased, alkaline phosphatase enzyme increased, especially in the alcoholic patients, which is causes damage to the liver. And by this we just end the seventh lecture of clinical chemistry. Hope you understand anything, and the upcoming lecture, it also contains a very large amount of information.